Hi students, I hope you are excited for today. We have some pretty cool stuff to be learning. We are starting our chapter on the cell, and that includes the discovery of the cell, the current functions of the cell, and even the diversity of life that we can see in the cell all around us. So we're going to get started with kind of the history and what we know about cells in general. The first thing we need to think about is how big is a cell? Cells range in size. You can see that starting here at about 100 micrometers, that's where most plant and animal cells are. We can have smaller cells. Bacteria cells are about in the 1 to 10 micrometer area, and viruses can be as small as 100 nanometers. Those are very, very small. Now remember, we talked about, is a virus really a cell? It's definitely a phospholipid bilayer coding some proteins, definitely has some genetic information in it, but is it a cell? Remember, we said that's kind of a gray area amongst biologists within their kind of descriptions and qualifications for what a cell might be. But virus, for sake of argument, is the smallest version of a cell. Bacteria are larger, but then plant and animal cells, that's us, right? We're the animal cells. They can be larger at 100 micrometers. Plant and animal cells are able to be visualized using a microscope. So you can look through a light microscope and see not only the cell, but also some of the interior structures. Not much, but you can see some of the interior structures. Chloroplasts are reasonably visible. The nuclei can be seen, but for the most part, we're not going to see a lot of interior workings of the cell with a light microscope. The light microscope and its resolution is up here in blue. The human eye, or no microscope, its resolution is in this kind of salmon color. So you can see that right here at the end of human eye resolution is where we find the cell. Humans cannot see a cell unaided unless it's a very, very large cell. And animals make really, really large cells as their eggs, egg cells. So frog eggs or salmon roe, I mean, if you eat poke, if you eat sushi, then you're probably familiar with like caviar, a little bit of that like kind of pink poppy things that are on the top of your poke, and those are actually fish eggs. So we can see those, but they are still large in comparison to other cells. The things that we were studying before, small molecules, lipids, and proteins, they're way down here at the far reaches of the electron microscope. Even proteins are pretty difficult to be seen with the electron microscope. So cells need to be a particular size. They can't be too small because they're going to need to hold all of the stuff that goes in them. And they can't be too big because if they get too big, a single cell gets too big, then it's hard for the cell to maintain its interior boundaries. There are so many happenings, there are so many things that need to be taken care of inside the cell that it kind of gets overtaxed. So cells have found this really nice balance between being too small, big enough that they can hold all important pieces that they need to function, but also not being so big that it starts to become difficult for them to maintain and keep their interiors healthy. So you might be wondering how we know all of this. Well, in the 1600s, Robert Hooke first invented a light microscope, the first one, where he used bouncing wavelengths of light off of mirrors, and he created a way to see with high magnification. Once he saw with a light microscope, one of the first things he saw was cork cells. Now these are dead cells. This is not a living thing. Cork, like what holds a wine bottle closed, it's really just bark. It's kind of like a tree bark. And so what he's looking at here, what you're seeing, are these little cell walls. We're going to talk about the cell wall later and the fact that plants have cell walls, but we're looking at these little cell walls, and so they look like the little rooms in a monastery. If you go to a monastery, the monks live in these very small, repetitive rooms. And so he thought they looked like the cells of a monastery, and that's how he named them. As time passed, our microscopes got better and better and better, obviously. And so as we do, the idea of a cell goes from the more primitive concepts that Robert Hooke had to what we now consider the cell theory. The cell theory has 3 points, and you should make sure you write them down and you know them, and you're able to identify them for a test. So the cell theory says that all living things are made up of cells. So if something is alive, it must be made of one or more cells. So you and I, we're made of many cells. The grass outside, the trees, those are made of cells too. Fish, dogs, cats, all made of cells. Mushrooms, made of cells. Anything that is alive. If it's not alive, it may or may not be made of cells. Maybe it was once alive, like a piece of furniture maybe is made out of wood, and you might be able to find some cells in that wood. But other things were never alive, like plastic. When you look in the interior of plastic, you're looking at a material, at matter, but not at a cell. So that way you would not expect to find cells in something like plastic. Cells are the basic unit of structure and function of living things. So not only are living things made of cells, but they're made of pretty much only cells. That's what that second one means. Not just they have cells, like they have 2 or 3 cells, but they are made of cells. They are completely full, chock full of cells. And the last piece of cell theory says that all new cells are made from old cells. They are reproduced. So we will not have what they call spontaneous generation. If I were to just have my plastic sitting here in front of me on my table and I let it sit here, I would not at some point find cells. Or if I did find cells at some point in here, I would know that that was because there were already cells on the table. Maybe there's already bacteria here. So if I leave it long enough, I might see bacteria start to grow. Well, those are just cells coming from pre-existing cells. They're not appearing from nowhere. And the original kind of thought was that you would see like a steak or a piece of meat that was left out. And if you left it for a while, it would eventually get maggots. It's disgusting, right? And maggots are just fly larvae, but they didn't necessarily know that. And so the question was, how did the meat get maggots on it in the first place? And of course the answer is, I hope you know, that the flies would come and land on it and they would lay their eggs in the meat. Those eggs would eventually hatch and become larvae. So the larva wasn't coming from nowhere. It wasn't spontaneously generating from the meat. It was incubating in the meat and then hatching into larva. It's a gross story, but it does explain where that kind of question was coming from. Like, how do we know if that larva just spontaneously comes from nothing or if it came from something else? Of course, this brings into the question, well, but then how did you get the initial cell? And spoiler alert, we're going to spend time talking about what would have been required for a cell to emerge, the first cell or the first cells. And basically, like, it kind of boils down to, we can't have oxygen in the atmosphere. We have too much oxygen in our atmosphere to ever even believe that we could see a cell come from non-living matter. Back when cells first evolved, or when it's proposed that cells first evolved, there was not oxygen in the atmosphere. There was an entirely different atmosphere at that time, and that atmosphere could have been conducive to the first life generating, but not the atmosphere that we live in anymore. Oxygen is a big bully. Oxygen is bad for spontaneous generation. So how do we know any of this? We study using light microscopes. Light microscopes use a series of mirrors and beams of light to magnify an object. And so a light microscope like this one can magnify an object up to 1,000 times. There is one magnifier ocular lens up here that magnifies times 10. And then there are usually 3 different objective lenses down on the nose piece. So you can zoom in as you go, and the highest one being at 100 times. So 100 times 10 is 1,000. And so that would be the magnification, the maximum magnification that you would be able to get. Using a light microscope to look at an Elodia leaf, this is the kind of plant that you put in your aquarium. If you have an aquarium, this is the kind of plant that you put in there to oxygenate the water. Looking at an Elodia leaf, we can see the cell walls around the outside, the cells, as Robert Hooke termed them. And then these little green dots in there are chloroplasts, and chloroplasts are where the light energy becomes sugar. We're going to talk about that later. My point, though, is that I want you to notice those little chloroplasts in there, these little chloroplasts, because on the next slide, we're going to look instead at what these chloroplasts look like using an even stronger microscope, an electron microscope. So the benefit of the light microscope is that the things you're looking at can be alive. In an electron microscope, they can't be. But the drawback is that in a light microscope, this is as clear as we can possibly see our chloroplasts. It's not very clear. In an electron microscope, I'm going to skip ahead now. In an electron microscope, this interior, these little folds right here, these back and forth kind of accordion folds, those are a chloroplast. And so under an electron microscope, we are seeing with much greater detail, resolution, and magnification the cells that we are looking at. There are 2 kinds of electron microscopes. The first kind is called a scanning electron microscope, or an SEM, scanning electron microscope, and it runs along the surface of an object. So this was a scanning electron micrograph of a bug that was found living on a daddy longlegs, on the body of a daddy longlegs. So picture a daddy longlegs spider, right? And now imagine that like teeny tiny body that they have, and then identify that this bug, this like patchy skin right here, that's the skin of the body of the daddy longlegs spider. So this is a really lot of magnification on something that we can kind of visualize. These ones are, instead of scanning, these are transmission. So these ones are looking through a specimen that has been sliced really thin, like a deli slicer. Like imagine looking through a piece of deli meat. That's what they're looking at. And so right here, this is a mitochondria, and you can see all the folds and the cristae membrane inside of that mitochondria, all magnified up. So we can obviously see a lot more detail here. The drawback to the electron microscope is that anything you look at with an electron microscope is dead. It needs to be dead, preserved, studied, and then as the electrons bombard it, that's what the electron microscope does, it bombards it with electrons, it burns up the specimen. And so you can really only study it for a very, very short period of time. I've actually done some work on an electron microscope before, and it is pretty difficult to get a good picture, and you definitely can't reproduce the picture. By the time you snap the picture, you have actually lost the specimen. The specimen has been destroyed as you were using the microscope to get your visual. There is a brand new technology. It's brand new from the 1990s, which doesn't feel brand new, but it's pretty new when you think about creating the technology and then also getting it out there. And so this is called a scanning probe microscope, and it allows you to study small things like an electron microscope, but you can do it live. And so this has been very helpful in studying living systems. We want to see things as they're working alive. So this is a scanning probe micrograph of a Plasmodium infected red blood cell. So Plasmodium malariae, this is a human of malaria, and part of its life cycle, it lives inside of a red blood cell. Now, I know this doesn't look red. First of all, red blood cells aren't red when they're in your body, unless they're attached to oxygen, so they don't have to be red. But secondly, they have to recolor everything, and so they recolored it, and it's yellow. But this is a cell that is fully bursting with malaria parasite protein clusters that are on the host cell. And so you can see the little clusters. That shows you that the cell has been infected. This would be something that would be useful to find out, the number of infected cells in a person. You'd be able to look at their blood and determine whether or not they were infected or how far along the infection was. But in general, scientists aren't great at using them yet, so they are potentially very useful, if not immediately useful. So when we're studying cells, there are really two kinds that we're going to be looking at, those that are prokaryotic and those that are eukaryotic. Prokaryotic does not have a nucleus, although they do have DNA in circular bundles called plasmids. They have only DNA, ribosomes, and a cell membrane or a cell wall, or both. Bacteria are prokaryotic. Also, another group called archaea that you maybe are not familiar with are considered prokaryotic. So these are both kinds of cells that are prokaryotes. They do not have a true nucleus. They don't have very many organelles, and they're very, very small. The eukaryotic cell does have a true nucleus. The eukaryotic cell is the animal cell. If in your grade school you studied the cell and the cell had a nucleus, then you studied eukaryotic cells. So they have a true nucleus, they have many organelles, they're very large, they have a membrane, and they can have a cell wall. And these eukaryotic cells, because of their size and also their intricacy, can make multicellular organisms. So we're going to be looking at both of these groups of cells, but we're going to be really focusing on the eukaryotic cell. Even there's way more prokaryotic organisms than there are eukaryotic, but in terms of interest, we're pretty egocentric, and we like to study us, and we are eukaryotic cells. So we are going to be focusing on not only eukaryotic cells, but animal eukaryotic cells. They're 100% our cells. We will be spending our time studying the eukaryotic cell from both the plant and the animal. But I just wanted to take a moment and show you some of the differences. So this is a prokaryotic cell here. It has only a nucleoid region. That's where it stores its DNA. But the DNA is not protected. If the DNA was protected, we would call it a nucleus. So now you already know what the function of the nucleus is, right? Protect the DNA. A prokaryote does not have a nucleus, just a nucleoid region. It will have some ribosomes. Ribosomes make proteins, so you can't have any cells without ribosomes. So it would have its ribosomes there, and then it's going to have some coverings to protect it and keep it safe. And in short, that's everything that's in a prokaryotic cell. Now, a eukaryotic cell takes those things and it adds a whole bunch of layers of intricacy and diversity and unique organelle features in order to make the eukaryotic cell work the way that it does. Eukaryotic cells form all of our eukaryotic kingdoms. So that would be our protista, our animals, our fungus, and our plants are all going to be based off of this same individual eukaryotic cell, unicellular or multicellular. So next up, we're going to be talking about the structure of the animal cell, and we will start with more of the intricate, unique specifics of the animal cell tomorrow in class.